So with that, it's my pleasure to introduce our next speaker, Jean-Baka Domelevo Entfellner, who is a bioinformatician at the International Livestock Research Institute in Kenya, where he's head of data analysis and research. And he leads the Bioinformatics platform. But in addition, has been designing and implementing some really creative and exciting capacity building initiatives across Africa, from Cameroon to Niger to Kenya, and even I think in South Africa. So really excited to hear about some of this work. And he was also part of the Lab genome sequencing project, which you heard about from Bernice. So I think he's going to tell us a bit about that, as well as the capacity building work. And with that, I'll let you take it away, Jean-Baka. Thank you so much.
Thank you very much, Rose. Can you hear me well?
Yep, we can hear you.
Excellent. Okay, let me start sharing. It's fine as well. Okay, can you see my screen?
We can.
Okay, excellent. Thank you so much for the introduction. So as you said, my name is Jean-Baka Domelevo Entfellner. It's not an easy one to pronounce. I'm working in Nairobi, Kenya since 2017. I had the Data and Research Methods unit. And prior to leading that unit, I was the head of bioinformatics at the ILRI Hub, one program within ILRI. So I'm going to talk a bit about the communities of practice in genomics and bioinformatics, two cohorts of more or less the same model for capacity building in Africa. And here in the picture, you can see Bernice, who just talked to you right now. So let's go. The outline will be the following. I'll talk about the African context, then specifically our training programs, the outcomes we had, and then we'll discuss, and I guess the discussion will be prolonged into the panel discussion after that. So let me talk a little bit about the context in Africa, a few statistics just to give the big picture. First, the expenditure in research and development is extremely low on the continent. So here in this graph, I show data from the World Bank showing on the y-axis the research and development expenditure, that's percentage of GDP, and that includes the public and the private sector. And you see two groups. At the bottom, we have the African nations. Don't ask me about the specific pick of countries. It's just because some countries do not report properly to the UN stats division, and so they are not in here. And sometimes you'll see from one graph to another, we won't have the same selection of countries. So sorry for that. And on top, you have the so-called developed countries, mostly in Europe or in Southeast Asia, like Singapore. It's interesting in this graph to see that China has walked the journey of a country in the bottom of that graph to a country that sits now in the top with the industrialized or advanced economies. So that was about the expenditure, how much money is spent on research and development in a country. And you see that African countries remain below the 1% mark because, of course, having other priorities to deal with. That's the workforce now in terms of researchers, again, in public sector, universities, etc., but also in the private sector, researchers in research and development. And be careful here, the logarithmic scale applies on the y-axis. So you really have tremendous differences in terms of the number of people that can participate in the effort to create knowledge and develop this knowledge into practical applications between, at the bottom, the African countries, Madagascar, Congo, Togo, Niger, Lesotho, for instance, and at the top, the most advanced economies, and in the middle, we have a group of BRICS, and in that you have Egypt in this case, specific case. The good thing is that the continent is very promising in terms of its demographics. It's both a good thing and sometimes an issue, of course. But we have a very young population. In this graph, I plot the dependency ratio. That's the proportion of dependents aged less than 15 years old. This ratio to the population of working age people. And sometimes the ratio is even above 100%. In Zambia, so on top you have the African countries. Again, we see one country that walks its way from the group on top here to the group at the bottom, that's China. And we see also Singapore having a real problem in terms of its fertility rates. But so the African population is still predominantly a young population. I see lots of young entrepreneurs. It's a very innovative continent. You can see we have very good fintechs and mobile money on mobile phones that don't need to be a smartphone. That technology actually was born in Kenya with an operator called Safaricom. So it's a very promising continent in terms of its dynamism, and that's partly thanks to its young population. I have here shown some recent initiatives on the continent that shed some, that give some hope, let's say. In Africa, you have some countries that are pushing the budgets in research and development. Especially Rwanda has been kind of one of the good pupils on the continent, launching some initiatives, some institutes, etc. And Kenya is also one. So you have a number of initiatives that exist about doing better in science and in higher education. And that's of course a good thing. We have Africa CDC that is now an efficient or a structure that is akin to the American CDC, and that starts to gain traction in the wake of the global pandemic, of course. Opportunities for low and middle income countries, opportunities from genomics. I guess perhaps you've seen this slide from Bernice, but I wanted to insist that we have a wealth of diversity in our crops, but also in our other species like livestock or species close to livestock with a very interesting genetic makeup explaining the higher levels of resilience to drought, extreme heat, etc. So orphan crops or neglected, understudied crops or forgotten crops, and some of them that need a bit of research, that's especially the case for grass pea, need a bit of research to become really useful to the population. So we need to combat some toxigenic effects or genes. And the goal is, of course, to reduce the dependency of the continent on imports, especially on food. An interesting opportunity also comes from the technical side. We have now more affordable genomics platforms. And this is an example here I'm showing, the Oxford Nanopore Menion, I'm sure you're all familiar with. This was used in the field for the cassava virus action project. And it has low cost, no maintenance is needed, minimal power and temperature requirements. And also you have a flexible throughput, contrary to the big platforms you all know from Illumina and PacBio, for instance, where it's not flexible at all. You have to have the volumes of samples to be efficient in terms of costs. The unique advantage of the Oxford Nanopore platform is the ability to wash and reuse a flow cell. So it's good for small projects as well as for larger ones. So that was just a little bit to show the context in which we design our training programs at the Beka ILRI Hub. And we designed community-focused capacity building initiatives. We call them community of practice. Actually, we figured out that perhaps we should have called them, or we should move to communities of learning or communities of experiential learning, because it's about learning through experience. And at the bottom here, a few, some of the people who were most involved in the design of the first of these two bioinformatics communities of practice. So the idea was to stop with the kind of very short trainings that sprinkle some knowledge and that are very often used to justify activities of what we call predatory science, or a friend of mine uses the expression mosquito research or mosquito science in Africa. The mosquito comes on your skin, just takes a blood meal and goes away. So we've had this story of collaborations that were uneven because collaborators from advanced countries come, just take samples, benefit from the knowledge, the local knowledge of samples of interest, and then all the research is done outside. So that's not what we wanted, and we wanted to create communities of practice deeply rooted in Africa and meant to empower African scientists with the skills necessary to tap into the great opportunities from genomics and bioinformatics. So we decided to have an in-country long training, and the idea was to focus on a few people instead of training for 2 days, 50 people. We said, okay, let's just train a few for a longer period of time, and it will be more impactful once these go back to their home countries. The idea of peer learning was very important. So you'll see we really insist on the word community. We created a community drawing early career scientists from different countries on the continent. They came to Nairobi, worked together, learned together, struggled together to master some concepts and some techniques, and learned from each other. So the peer learning is a very important aspect of our model. We had two such cohorts of the bioinformatics community of practice. The first one started in 2018. We had 14 fellows from 8 countries. We had quite a bit of a gender imbalance. We had only 4 female trainees for 10 male. It was an 8-month residential training to start with 7 training modules and 3 mini projects. I'm going to show you. The training model was in 3 phases. So first, we build the fundamental skills through classical lectures and also periods of exercises. I'll get back to that. Then we empower the trainees with additional skills, leadership skills, soft skills, etc., so that they are in a better shape to then amplify the impact of the training by training in turn within their regions of origin. And throughout this training, we really encouraged peer learning, continuous interaction as a community. The first phase called Build, so that was between April and October 2018, 7 to 8 month residential training, and we had all these fundamental modules for bioinformatics, for genomics. Most of the trainees didn't have a good experience with, didn't have a prior experience with shared Unix environments, programming, etc. So the first modules were really much on this, and then we had some on variant calling, quantitative genetics, etc. Key to this was that we had the budget to have some trainers come in, various trainers came in, and also tutors were involved. It was important for us to have not only weeks of formal lectures, but to also have weeks during which the trainees were working together on exercises to directly apply the knowledge gained. So many tutors were involved, for instance, PhD students from UK universities from our partners in this project. The second phase was called Empower. We had a train the trainer idea behind the fact that we wanted to have as many as possible of our trainees register in the carpentry trainings. We also had our trainees act as trainers and facilitated training in art at ILRI for others. And we had a workshop organized on soft skills. Phase 3, the amplify phase was when we had a mature community able to translate the training they gained into actually a kind of a ripple effect on their regions. So we organized regional trainings in 2 different locations, Addis Ababa in Ethiopia and Bukavu in DRC. Those trainings were hosted, facilitated by the fellows, so we had really trainees becoming trainers. We developed also online tutorials and videos. Here you have 2 snapshots. One from a video where Bernice was featuring, and it was online video, an online blast, and another one where Muihaki, another colleague, was talking about how to avoid predatory conferences, because we recognize that some of these skills about using tools or about being aware about which journal to publish to or which conference to go to. Some of these skills are missing or are difficult to get for some people in Africa, or far from the kind of knowledge we can have in other places about journals, etc. And of course, this phase, actually, I said 2019 plus, because it continued way beyond the end of the formal funded project. And we have 2 strong whole genome sequencing research projects, one on African yam bean that is fully driven by the group, and one on lab bean genomics, where we heavily participated, and Bernice talked about that. In terms of funding, it wasn't a huge funding to do that for so many months. So I believe we maximized the impact we could have with the money we were given. So it was a collaboration between the ILRI Hub with the funding from the Africa Biosciences Challenge Fund and the GCRF STARS. That was a program by the BBSRC. The British Research Council on Biological Sciences. And these were my 2 co-investigators, Christopher Wawi from JIC, the Joint Center in the UK, and Rob Davey from the Earlham Institute, also in the UK. The second cohort was a slightly different one. We had less money from the BBSRC following the cuts on the ODA funding by the UK, but we also had in-kind funding from Oxford Nanopore Technologies. It was post-COVID, so we had to postpone it. We got a better gender balance, so still fellows from various countries, and a better gender balance of 7 female to 8 male. So because of the lack of funding, we shrunk the training, but still to something significantly longer than the 1 week workshop on this or that. So it was a 3-month residential one. And we had a focus this time on 3rd generation genomics and the bioinformatics analysis for long reads. And 3 mini projects, one long-term genomic study that Bernice talked about a little bit on the Vialis-CAFRA. The outcomes to finish. So, of course, a paper that was published recently on the chromosome level genome assembly for the lab bean. And we hope to publish soon our African yam bean assembly, also chromosome level. I mentioned also here that young Kenyans, among which you have David Kiragumora, one of the trainees in the second COP, who started the Bioinformatics Hub of Kenya. So it's a hub of young enthusiasts who are working as masters or PhD students, or sometimes employed or sometimes unemployed, who say they need to develop bioinformatics, and they are really deserving because they go with so they perform outreach actions. They go to universities, and in Kenya you have many universities, but many universities away from Nairobi where no one has ever heard about bioinformatics. So they go and they talk about the opportunities in genomics and bioinformatics, and hopefully they will help more universities offering programs in bioinformatics in Kenya, in Uganda, in Tanzania. We also have the ENBIT regional network for bioinformatics training. I'm part of that, so I'm very happy to be part of that. And that's another initiative that was kind of a sister initiative, but just focusing this one on actually having universities embrace bioinformatics. And so the ENBIT made it that one university on the coast of Kenya at Kilifi designed its curriculum in bioinformatics at master's level, and the same at Makerere University in Uganda. The fellows now are all occupying prominent positions, some still studying abroad in Africa, others contributing in their countries to the regional or continental capacity. We have Ahadi, for instance. He was appointed as regional coordinator in the fight against SARS-CoV-2 at the moment when that happened, and now has a collaboration with TIWAS in Italy. We have Bernice, who talked to you, and now she's a PhD fellow at John Innes. Some examples here. I'm talking to you from Dakar, Senegal, where I'm delivering a training, and I met with Idrissa. Now he has secured a position, a tenured position at Pasteur Institute of Senegal, and they work on having a portable disease diagnostic lab also based on Oxford Nanopore technologies. So all very promising young scientists, and most of them are working, if not in Africa, closely with African colleagues. We run a post-training survey, and this on the left is a word cloud for the best of the trainees' experience, and on the right the worst. So time is never enough, and they said that they wished they could have had more time to learn more, but they really enjoyed the experience and the group aspect, the fact that they formed a community, a team, and they say. Finally, don't hesitate to tell me if I'm late, running late, discussion quickly. So what worked? The longer residential training is a very important aspect, longer than the short courses that are too easy to offer in a way, and easy to tick boxes for funding agencies. Okay, we trained 50 people, etc. But what remains from this knowledge that was sprinkled on people for a very short amount of time is really questionable. Peer learning works very well, and now we still see that papers are coming from our students who keep on working together, our fellows who keep on working together. Challenges the funding, so the funding stopped from the BBSRC, and in any case, funding model is not fully sustainable if it's money coming from outside Africa, and that's why I have 2 bullet points on the right about that. It was difficult also to maintain sustained interaction. So that's why the AYB project took so long, because it's difficult to meet every 2 weeks and keep on working on a project while you have your other tasks on your desk. And limited opportunities within Africa still encourage somehow some brain drain, but we want to reverse that. And very often now we see scientists coming back to Africa after having a PhD, having gained a PhD outside. Way forward, I'm just going to talk about the 2 bullet points. Really, it's important for us to encourage African nations to spend more on their research and development, understanding that if we want to tap into genomics, if we want to solve problems for the future about securing affordable food in a context of climate change, for instance, no one outside of Africa will take any interest in studying crops and species and pathogens that are of interest exclusively to Africa. It's easy to compare the budget spent on malaria versus the budget spent on other diseases or ailments that are more present in Europe and in the US. Advocate for a stronger, more focused African Union is important, and no one else can do that if not us Africans. The take-home message is slightly different from Bernice's. Really important to see that what worked in that path is that our students have shared goals from common interest in studied species and pathogens, their own. Okay, so the output we can get is not only, of course, the most important output is actually the capacity building, is actually having a better human resource equipped with the skills for genomics and bioinformatics, but also we want to show that Africa is able to generate its own chromosome scale, or actually telomere to telomere genomes, sequenced, assembled, analyzed in Africa. There is no hurdle to that. We have the technical capacity, and we have, if we have the manpower, there is no reason why we cannot do that within Africa. I think we demonstrated that. That's my last slide. Thank you. More people than these are to be thanked, and I added also Ian McLaren, who has been supporting us from Oxford Nanopore Technologies, and my very good friend Oluwaseyi Sorinola was instrumental in working with me to actually implement these training operations here in Nairobi, Kenya. I thank you for your attention.
Thank you so much. Will's going to introduce our last speaker.
Yep, thank you so much, Jean-Baka, for a really cool example of local capacity building and investment into Africa's future leaders.